Good afternoon. Welcome to Academia Sinica. My name is Chi-Hao Lee. I am the director and the faculty member of Genomic Research Center. I will be the moderator of today's special lecture. We have a full house today. Before we begin, I would like to take this opportunity to thank the International Peace Foundation, the Department of International Affairs, of course, Dr. Sir Paul Nurse for making this event possible. Before the lecture, the president of Academia Sinica, Dr. James Liao, will deliver an open remark. Please join me in welcoming President Liao.
Good afternoon, ladies and gentlemen. It's my great pleasure to welcome you to this bridge program. This is probably the sixth lecture at Academia Sinica. And this year, as you know, Peace Foundation has sponsored more than 30 Nobel Laureates to visit Taiwan within a short span of 6 months. And this is the sixth event in Academia Sinica. Of course, there are many other events in universities. And today is our great pleasure to have invited Paul Nurse, who is a cell biologist, a geneticist, who discovered the cell cycle regulatory proteins. I think most of you, or probably all of you who studied cell biology will know his contribution. So he received Nobel Prize in 2001 because of this groundbreaking discovery. And Paul is a very special person because he's one of the very few scientists, very few top scientists, who also are very good administrator or academic leader. So after he received a Nobel Prize, he also served as the president of Rockefeller University. And then in 2010, I believe, to 2015, he served as his first term as the president of Royal Society in London. And during that time, he set up the Francis Crick Institute. Later, he served as the president of Francis Crick Institute after he finished his serving as the president of Royal Society. And more especially, Royal Society recalled him only a few months ago to serve as the second term as the president of Royal Society, which is completely unprecedented. Okay, the Royal Society presidency can only serve 5 years each term. And he served 2 terms separated by about 10 years. So he assumed this new position only 2 months ago. And we, Academia Sinica and Royal Society, have very close interactions in the past few years. We have held joint symposium twice, each time in Taipei and 1 time in London. And we finished our second time only a few months ago, and we are going to plan for the next one. So today we are very pleased to have Paul Nurse to give our lecture. I'm sure you'll all enjoy his wonderful lecture. Thank you very much.
Okay, I prepared some notes, but I think President Liao has already said most of it. I'll make it short. It is with great pleasure that I introduce today's speaker to you, Dr. Sir Paul Nurse, who I think many of us consider him is one of the architects of modern cell biology. And he has devoted his research career in understanding how the living system organize, maintain order, reproduce, and evolve. If you don't know him before, I'm pretty sure you must have read his finding in the textbook, particularly in cell biology. So Dr. Nurse studied biology at the University of Birmingham, and he obtained his PhD in University of East Anglia, and then he conducted his postdoc research in University of Edinburgh, where he began his work on a simple but very important question: how does a cell divide? Using a very simple, maybe that's not the right word, use the fish is a model organism. Dr. Nurse has made a series of discovery that revealed the molecular mechanism governing cell division. He has identified a key gene CDC2, or cell division cycle 2, which we now call a cycle-dependent kinase. This protein is very important in the regulation of the progression of cell cycle from the G1 phase to the S phase, and then from G2 to mitosis. His lab later demonstrated that this regulatory mechanism is conserved from yeast to human, thereby establishing that a simple and ancient machinery regulate the cell cell division across the eukaryotic life. As President Liao had just mentioned, for this seminal discovery, he is shared the 2001 Nobel Prize together with in physiology and medicine, together with Lina Howell and Tin Han for the discovery of important regulator in cell cycle. So I think in preparation of this introduction, I stumbled upon Dr. Nurse's biography from the Nobel Prize website. Dr. Nurse described how he became interested in life science at a very young age, and his journey to accomplish all his many major accomplishments. I found it very inspiring. I encourage all of you to read it. I want to share something he wrote about his PhD experience with particularly the graduate student in the audience. Here he goes. Like many students, I found the drudgery of real experiment and the slowness of progress completely shocked. And at my low points, I contemplated other alternative careers. I'm pretty sure many of us have the same similar thought at some point. But this experience did teach me the need for sympathetic support of scientific colleagues, because at the forefront of research, there are so many difficulties that depression and low motivation are a constant danger. He continued to write, which I think is very important now only for the graduate student, but for all of us. I felt strongly that since pursuit of good science was so difficult, it was essential that the problem being studied was an important one to justify the effort expended. So for the struggling graduate student in the audience, there is a hope. You may one day win the Nobel Prize if you are asking the right question. So today, in his lecture, What is Life?, Dr. Nurse invited us to step back and reflect on fundamental principles that define living systems.
Thank you. Thank you very much for those very kind introductions. It's a pleasure to be speaking today for the Peace Foundation. I just wanted to say something about that, because I think education and research, and particularly science, is a way to break down barriers between people. It's a common language, the pursuit of knowledge. And this, I think, is the central core to the Peace Foundation, and I very much appreciate the efforts that have put together this program, of which I'm part, to actually deliver that. I'm going to talk today not about my own work. I'm going to talk about one of the more, perhaps the most fundamental question in biology, which is, what is life? It's an easy question to ask. I have to tell you, it is not so easy to answer. And I have to admit to you that by the end of it, you won't get a complete answer. If you're disappointed about that, you can always leave now if you wish. But I hope I will give some indication as to how you should think about it. Life. There's an extraordinary diversity of life on our planet. All around us are bacteria, fungi, plants, animals, our cells, human beings. And what is it that links all these different life forms together? It's a long time ago, but I think it was a butterfly, like this one here, that first started me thinking seriously about questions like this. It was early spring. I was about 12 years of age. I was sitting in the garden in northwest London when a quivering yellow butterfly flew over the fence. This is that yellow butterfly. It's a brimstone yellow. It turned, it hovered, briefly settled, a shadow disturbed it. It took flight again, disappearing over the opposite fence. Not a very exciting incident, you might say. But that intricate, perfectly formed, brimstone yellow, that's what it was, butterfly, made me think. Because it was both utterly different to me, of course, yet somehow familiar too. Like me, it was alive. It could move, it could sense, it could respond, it could develop into a complex living thing. It seemed full of purpose. And I found myself wondering, what does it really mean to be alive? Which is, of course, what is life? Now, I'm not the first person to have asked this question. For example, this is the title page of a book, one of quite a number of books with the same title, written by different authors, called What is Life? This one was written in 1944 by the famous physicist Erwin Schrödinger. His book was influential, including for me when I first read it now 50 years ago. His approach was to focus on genetics, and I'm going to say something about that. But my approach is going to be wider than that. What I'm going to do this afternoon is to describe to you, for me, the 5 most important ideas of biology, describing where they came from, explaining what they are, and then pulling them together to develop principles fundamental to life. The first of these ideas or concepts is the cell, the concept of the cell. The cell is the basic structural and functional unit of life. It is, if you like, life's atom. Cells were discovered by Robert Hooke in the 1660s in the city I live, in Oxford. There's a plaque on the wall, a very small plaque, but a plaque on the wall describing the first cells that he observed there. And he used a microscope like the one that you see here on the left. Used a razor to cut slivers of cork from a plant, examined them under this early microscope. And what he saw were neat arrays of boxes. His illustration's the one on the left there, the one in black and white. He called them cells after the Latin word cella for small cubicle. And it's shown in this drawing, which was published in 1665 in a book called Micrographia. On the right, I have a very modern scanning electron micrograph of the same material. You see, it hasn't really advanced much in the 350 years or more that in between those two pictures. A few years later, the Dutch draper, Anthony van Leeuwenhoek, who was working in the Netherlands, in Holland, in the city of Delft, scraped between his teeth, put what he found there under a microscope, and discovered single-celled life. These were bacteria. He sent this drawing to the Royal Society in London. He actually sent 1,000 letters to the Royal Society in London in the second half of the 17th century. And this is one of the illustrations that he sent there. I've actually handled this piece of paper, I have to say. And these are the bacteria he saw under the microscope. Look at figure B, fig B to C to D. Obviously the bacteria was swimming about underneath the microscope. Doing a sort of loop-the-loop, as you can see there. He was very disturbed, apparently, by this discovery of living microorganisms between his teeth, because he was rather proud of his dental hygiene, and it rather disturbed him to discover life in between his teeth. He was also a close neighbour in Delft to the painter Vermeer, Johannes Vermeer. And unusually, for a painter of the time, or for that matter of any time, he painted two pictures of a scientist. This is one of them, shown here, a geographer and an astronomer in Frankfurt and in Paris. And I like to think this is Leeuwenhoek, I have to say. There isn't too much evidence for this, although when Vermeer died, Leeuwenhoek was the executor of his will, so it is not impossible that that is the case. Anyway, over the next 2 centuries, it became clear that all living organisms consist either of a single cell or a collection of cells. And this was well summarized by a rather serious-looking German zoologist called Theodor Schwann. You see his picture here. And he wrote in 1839, translated from the German, "We have seen that all organisms are composed of essentially like parts, namely of cells." This states really that cells are the structural unit of life, but they're also the basic functional unit of life. This was expressed by another German, Rudolf Virchow, a couple of decades later, the founder of pathology, in actual fact. Nice sentence, 1858. "Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life." In other words, cells exhibited the properties of life. He also said that all cells come from cells. He wasn't the first to say that, omnis cellula e cellula in Latin. And this means, of course, that cells arise through the division of pre-existing cells. They don't occur spontaneously. They arise from connections in cells. I'm going to refer, they arise from pre-existing cells. I'm going to talk a little bit more about that at the very end of my talk. And this is the process, as you heard in the introduction, that I myself research on. So cells are interesting. If I fail to convince you of that, let me show you this picture. It's a picture of an egg surrounded by sperms, a mammalian egg, and I'll remind all of you in this room that you were all once a single cell like this fertilized egg. All of you were once a single cell, so you ought to be sort of interested in cells, at least for that. Now, cells can also be considered the simplest entities which unambiguously exhibit the characteristics of life. There are some entities which are a little bit more intermediate. I'll touch on those as well. And this, of course, means understanding cells is key to understanding life. So that's why I'm referring prominently to them now. So a cell. This is a modern picture of a cell, artificially coloured. Cells are bounded physical entities separated from their environment, separated by a semi-permeable membrane, lipid. That separates them but allows communication with those surroundings. This separation allows order to be built up inside cells at the expense of increasing disorder outside cells. That's an important feature. It requires energy to do that, of course, because in this way life does not contravene the second law of thermodynamics, a law that often troubles physicists thinking about life, because life at first sight seems to contravene that law. Schrödinger did talk about it. He touched upon it here in the top of these sentences. He was, top sentence here, he was referring to an organism's astonishing gift of controlling a stream of order on itself, and thus escaping the decay into atomic chaos. Beautiful sentence, by the way. But what he was here of course, saying that it was escaping the second law of thermodynamics. Thinking about that led him to propose that life had a code script. This quote, the second quote, is shown here. He was referring to chromosomes, more of those in a moment. These chromosomes contain in some kind of code script the entire pattern of the individual's future development and function. In other words, inheritance was responsible for the escape from the second law of thermodynamics, which is why Schrödinger put so much emphasis upon it. But this is an introduction to the second concept or idea that I have, the gene, explaining the mysterious phenomenon of inheritance. This is the idea of the gene as the basis of heredity. It had its origins with the work of the Augustinian monk Gregor Mendel, abbot of Brno monastery, then in the Austro-Hungarian Empire, now in the Czech Republic. And he published work on this in 1866. This is a picture of Gregor when he was abbot. Now, he tackled the problem of inheritance by carrying out crosses of pea plants which had different characteristics, analysing the outcomes in the thousands of progeny formed in these crosses. I found this picture of his garden in 1981 when I visited that monastery in the middle of the Cold War, actually, quite difficult to get to. And you can see here it was actually quite a big garden for doing these experiments. And he had a greenhouse of similar size. I mention this because this monastery was like a research institute, in actual fact. I mean, they were carrying out experiments in plant breeding, as you see here, but also maps of the moon, meteorology, and so on. It was a teaching order, of a Catholic teaching order, but it was also very experimental. And what Mendel did, he was trained as a physicist. I need to explain this. In the University of Vienna, he never passed his exams. Not all good scientists are good at passing exams. I'm typical of that, by the way. But he applied the thinking of physics, a quantitative thinking, to biology, which is a little unusual. And what Mendel did was cross peas of different characteristics, like the ones shown here. Plants, peas of different heights, different flower colours, different seed shapes, and so on. And he crossed them, and then counted very carefully the ratios of these different, what we call phenotypes, or qualities of the plants in the progeny. And these famously produced very fixed ratios like 3:1, and 9:3 to 3:1, and 1:2 to 1. And this led eventually to the idea of Mendelism. He didn't quite get to Mendelism. That took another a few decades. But that idea was that heredity was based on pairs of indivisible particles or factors. That's why they came out. It's like shuffling sort of marbles around. They could come out in fixed ratios. One of which is inherited through the germ cells, one from in animals, sperm and eggs, in plants from pollen and ovules. And it was the particulate nature that gave rise to these fixed ratios. Now, these factors were subsequently found to be linked to chromosomes that were discovered after Mendel's death. That's important too, because nobody took any notice of what Mendel presented. It just sunk from 1865 onwards, and was rediscovered in 1900 to 1901 by three other plant biologists. And they, there's a question of why was it taken seriously 30 years later. And I think one issue here was that in between chromosomes had been discovered. I've already mentioned them. And this allowed, this is a drawing from the 19th century showing chromosomes. And you see there, every dividing cell has got these strings, these chromosomes. And this turned Mendel's sort of abstract calculations on ratios into something concrete that you could actually see, so that the particles were linked with the behavior of the chromosomes. Now, these Mendelian hereditary factors are what we now call genes. in the 1940s, Oswald Avery in Rockefeller University, New York, where I worked for 8 years, showed that genes were made of deoxyribonucleic acid, or DNA, in mid-1940s. That also wasn't actually well received. He got a huge criticism for it, particularly from his colleagues at Rockefeller, because DNA was thought to be such a boring molecule that it couldn't be seen how it could encode so much information. This is a DNA molecule. Here we see the nucleus of the cell where the chromosomes are. Then the chromosomes and unraveled, and you have the famous double helical structure, which was established about 10 years later in London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, who did the experimental work, and in Cambridge by Francis Crick and Jim Watson. And Crick and Watson famously argued that the double helical structure of DNA could explain heredity through the linear sequence of bases making up DNA and the pairing between complementing bases, adenine pairing with thymine and cytosine with guanine. That's shown here. This is a double helix. It's sort of iconic structure, but it's easier to imagine if the helix is unwound, so you see it as a ladder. Imagine it just as a ladder with rungs going across the ladder. And the rungs are made up of links between those complementing bases, G with C, A with T. When these links are split, two templates are formed, and these can be used to make two new identical helices. And this explains how DNA is copied during cell reproduction, and it, of course, can account for heredity and also Schrödinger's code script. And similarly, it explains the permanence of the gene between generations, because DNA acts as Schrödinger's code script, precisely copied every time a cell divides. Crick went on to propose what's called the central dogma, explaining how DNA encodes the structure of proteins. Central dogma describes how the sequence of bases making up a gene is copied from DNA to ribonucleic acid, or RNA, that acts as messenger between DNA in the nucleus, where the chromosomes are, and the cytoplasm, where proteins are made. And there's one gene, in principle anyway, encoding each protein. These discoveries were fundamental. They laid the foundations for molecular biology. They'll be familiar almost certainly to everyone in this room. And they are critical for understanding the nature of life. Central to it is the genetic code. Heredity is written in a 4-letter code made of the 4 bases, A, T, C, and G, with the information stored in a linear form. This way of storing information is the same as the words I'm speaking to you, or sentences that you read in the book, or, for that matter, bytes in a computer. And it means the linear digital storage of information was invented by life over 3 billion years before the computer age, just to put it in perspective. Now, the nature and function of the gene, which is extraordinary, I think and I'm going to return to it, but it introduces the next 2 ideas, or concepts I want to explain to you: life as chemistry and life as information. First is life as chemistry. That is, how chemical activity and physical forces can bring about living activities. The origins of this idea can be traced back to Lavoisier, in fact, the father of chemistry. He was doing, he was of course a chemist, but he was interested in fermentation of wine, and was working out how fermentation could produce other products, and was thinking about how life could perhaps generate that. Unfortunately, his research career was cut short by the guillotine, because he fell foul of the revolution and was guillotined before he took his work very much further. But it was taken up by another Frenchman, also interested in fermentation, Louis Pasteur, who carried out experiments on sugar beet fermentation which was very important. He was analysing the fermentation of sugar beet being carried out by industrialists to make alcohol. And they were worried because the fermentations quite often failed. And instead of making alcohol, they made acid instead. And they couldn't sell acid, so this was commercially a problem. And they turned to Pasteur to ask what could be done about it. And he showed that to get a successful production of alcohol required the presence of a single-celled living organism, yeast. He proposed that the yeast cell produced substances that made alcohol from the sugar derived from the sugar beet. And if yeast was not there, and bacteria instead, then the fermentation made acid. So Pasteur concluded more generally that fermentation was a physiological act yielding products for the cell. Chemical reactions are an expression of the life of a cell. Excuse me, just getting a cough in my throat. In other words, what he was saying is life is chemistry, but it's a very elaborate and special chemistry based on carbon polymers, as are DNA and RNA. And I want now to consider carbon polymers that make up proteins. The substances from yeast that Pasteur postulated made the alcohol are proteins that act as enzymes which catalyze chemical reactions. Proteins are polymers of amino acids with a backbone and side chains of amino acids. The structure of part of such a polymer is shown here. The different amino acids have different chemistries. Some are positively charged, some negatively, some repel water, some are big, some are small. Examples shown here are 20 different amino acids that make up those proteins with different chemistries. The result of this is there's a wide variety of chemistry in proteins which can carry out many and varied chemical reactions. This contrasts with DNA, which is stable and is not chemically diverse. In fact, it's chemically rather dull. The chains of amino acids making up protein polymers fold up in many ways to make complex structures. You can see here a chain of amino acids beginning to fold, and they fold up in different ways because of their different chemistries. And they also then can fold up in a way like this billiard ball type of model here, where you have a three-dimensional structure with different chemistries going on in different parts of the molecule in principle. And what this means is that proteins can carry out an enormously diverse range of chemical reactions. They act as chemical machines, and these machines produce the huge range of chemicals required for living organisms to function. They make new molecules, they break molecules down, they recycle them, they generate molecular assemblies, they synthesize polymers of nucleic acid, lipid, and carbohydrate. They do physical work, they make motors that carry cargoes around the cell on protein-based trackways. They act in hybrid ways, combining chemistry with physics, capturing energy from sunlight to produce energy-rich chemicals, driving the activities throughout the cells. All this activity means that there are thousands of chemical reactions being carried out simultaneously and close to each other in living cells. A small sample of these are shown in these dots on this metabolic metabolism map. For those of you who are familiar, this is the Krebs cycle in the middle there. Can you see the red dots? Each of those dots is a different enzyme. Now I want to point something out which isn't perhaps emphasized enough. All of these reactions require somewhat different chemical conditions to produce the specific reaction. And that means they have to be separated one from another. And that means in turn that can only be achieved by compartmentation. Compartmentation that's brought about by the surfaces and folds of the proteins, of the enzymes, complexes of enzymes working together, and by membrane-bounded organelles, visible here in the cell. I showed you this picture before, but now I'm trying to emphasize this isn't a static picture. This is actually reflecting the many different chemical microenvironments within a cell that allows the hugely elaborate chemistry of life to take place. Now, for all of this to work together, you cannot actually keep these different reactions completely separate. They have to work together as a whole. It has to be organized together, which requires communication between the different chemical microenvironments and compartments. Communication is, of course, the transfer of information, and that leads to my next idea, life as information. Life is a complex system. This was first argued by the philosopher Immanuel Kant. He was I came across this in a book on moral philosophy, you won't believe it. He's got about 15 pages describing the complexity of life. This is around 1800. And the operations of this complex system is based on the management of information. And I want to give you two examples to illustrate this. The first is the structure of DNA. Of course, you've seen this. It's iconic, it's beautiful, but its real beauty doesn't actually really lie for me in its structure. It lies in the fact that it only makes sense when it's recognized as a digital information storage device. That's the real beauty of DNA. The one written in that linear script of 4 letters. It's ideal for storing information with the encoding letter bases turned inside the helix, which is protected from chemical change. This information, though, which is stable, chemically stable, and therefore uninteresting chemically, can be turned into chemical action that can do work through the varied chemical activities for proteins. And for me, this is the wonderful characteristic of life, all built on carbon-based polymers. The ability to encode information in inert linear structures in DNA, and then turn that into a protein structures that can carry out the chemical and physical work in active 3-dimensional structures. So you separate the storage of information away from the chemical work that has to be to keep the cell alive. Something, again, I think we don't emphasize enough. Second example about information I want to tell you about concerns regulation. This is making complex systems in living organisms act as a whole with purpose. Now, it's easier to turn to man-made machines to get hold of this. And I don't know if you're like me. I understand most machines that we've made before 1900, and increasingly not machines made after 1900. This one is a governor that controlled a steam engine, built in 1896, this one, in a steamship I was in New Zealand. And this is a governor developed by James Watt to regulate steam engines. Now, it's simple to understand. As the engine goes faster, those heavy balls swing out, and they lift up a valve, and they cut off steam going to the engine. As the motor slows down, the balls come back and provides extra steam back into the steam engine. And this maintains a constant speed of the steam engine without human intervention. It's an example of homeostasis, and it's, of course, a negative feedback loop, shown here in the top cartoon I have here in the context of a metabolic pathway in a cell. If we look at that, we've got A being turned into B into C, and C, as it builds up in concentration, inhibits A to B, and as a consequence, the level of C drops. So this is a negative feedback. It gives you homeostasis. Beneath it is a positive feedback, rather similar, A to B to C, but C speeds up A to B, so this acts as a switch. Once you've got to C, it is an irreversible switch. These types of feedback loops were first postulated by two brilliant French geneticists, François Jacob and Jacques Monod, studying simply gene regulation of sugar metabolism in bacteria. very little biochemistry or logical thinking and genetics, but a real example of genius in my views. Now, many such feedbacks and control modules operate in cells. They maintain homeostasis, they generate switches, as I've described, timers, toggle, oscillators, translating chemistry now into informational modules. So we're now going from chemistry into modules of information. And these can be linked together to generate even more elaborate routines of informational management and processing. A metaphor for all of this is an electronic circuit. This is an old-fashioned transistor radio, and I stole it from a journal. And for those of you who work in cell biology or cancer, you'll see the person writing this paper had put on the circuits here various enzymes that have been implicated in cancer and the control of cell division. This allows the system to act as a whole with overall purpose. If it's a radio, then of course it can pick up radio waves and produce voices and music. Or if it's in a living cell, it can reproduce a cell or make a butterfly or can maintain our cells. By the way, this is hardware, of course. But one thing I want to point out about cells is that a term that's been used by Dennis Bray describes what happens in cells as wetware rather than hardware. Why does he say wetware? Because you communicate from one module to another in a cell through solutions, aqueously, you connect. And that means you can very easily rewire it in different ways. And this provides a much richer way of managing information that we've barely actually touched upon, I'd say, at the present time. Now, for centuries, the generation of purposeful behaviors was thought to require a designer, a creator. And it took evolution by natural selection to change this, and this is my final and fifth idea, after which I'm going to try and summarize everything up for you. Evolution by natural selection, how living things come about. This is probably the most beautiful idea in biology. It's mainly due to Charles Darwin, not entirely, because the idea has two parts. The first is that life evolves, and evolves over geological time. And the second is a major mechanism for evolution is natural selection, which is largely Charles Darwin. The first was actually, that is, that life evolves, had been discussed for a while. And I'm going to introduce you to Charles Darwin's grandfather, Erasmus Darwin, and you see here on the left. Erasmus Darwin will be less familiar to you, and I'm going to spend 1 minute on Erasmus Darwin, because he was really a very interesting character. Evolution had been thought about in the 18th century by people like Erasmus, people like Jean-Baptiste Lamarck. Erasmus was a colourful character. He was a scientist, he was a doctor, he was a poet, and he wrote all his research up in poems. It's all poetry. I've even got some of his books in poetry. I mean, it seems ludicrous today, but at least something quite fun about it, I have to say. So it's all in poetry. He was a Republican. He refused to be the doctor of George III, and he was asked several times because he was very successful. He was an early proponent of women's public education and wrote a book on what teachers should teach women, and his two daughters set up a school publicly for women, and he was a great conversationalist, even though he had a terrible stammer. And he was nowhere near a university, nowhere near Oxford, nowhere near Cambridge, nowhere near any university. He was simply a country doctor, and a member of what was called the Lunar Society, which met once every month under the full moon, and included people like James Watt, and was in the Midlands and was the centre of the Industrial Revolution in the 18th century. I just like Erasmus. He's a bit more colourful than his son Charles. Now, back to Charles. The evolution of life, and Charles here is the one on the right here, of course, implies that there is a tree of life. That is, living organisms are related. If life only arose and survived once, as seems likely given the conserved chemistry and coding found in all living organisms, then all life on Earth is related to each other. That's a critical thing to remember. This connection of life was found in the only illustration that you find in Darwin's Origin of Species, published in 1859. This is the first tree of life, and you can see in the tree that organisms go extinct, and what we have at the top is the living organisms that still exist. And look at the bottom there. You've got A to L. He wasn't sure whether life arose more than once, or perhaps many times, but maybe only survived one particular a part of life. So he has it from A to I. And what I just said, of course, is given the similar chemistries, maybe, although it may have had multiple origins, it would have only survived once, because the chemistries are similar. Darwin gathered his evidence for this through his trip around the world in a little boat, a naval boat called the Beagle. Now, his idea of natural selection is a consequence of populations of a species exhibiting variations that are caused by inheritable genetic changes. These variations can be caused by mistakes when DNA is copied, or if DNA is damaged by external causes. If these variants influence characteristics that make those organisms more successful, better adapted, then they will reproduce more, and that particular genetic variation will spread through the population. Over time, variation can accumulate and lead to changes in the population such that the individuals of the new population can no longer breed with the original population, accounting for the title of his book, The Origin of Species. It's the origin of species that you see there. And that is better adapted and more successful in survival and reproduction. Darwin came to this view in part by his study of finches on the Galapagos Islands. You'll see on the left there the different beaks of the finches that he actually described, which are adapted for different purposes. At the top we have clearly a beak that is good for cracking nuts. At the bottom you have clearly a beak that is good for getting insects underneath the bark of a tree. These different finches become adapted to their lifestyle, their environment, by being specialized better for what they have to do if they are to succeed. Just like the different pliers on the right here are specialized to do different jobs, but actually, of course, have a similar structure. But of course, the difference between beaks and pliers is that pliers are intelligently designed by human beings for different specialized purposes, whilst the finch's beaks achieve specialized purposes through natural selection without a designer and without a creator. And this is, of course, is the reason for the continuing conflict of creationists versus evolutionists. Now, for natural selection to take place, you need three things: reproduction, a hereditary system, and a hereditary system that exhibits variability upon which selection can work. And I'm going to pull all of this together with the ideas of the cell and the gene shown here. Imagine a single-celled life form with a brown cell coat, the color of which is determined by a gene. Imagine when the cell copies its DNA, it makes a mistake. So a cell coat gene is altered, and the cell now makes a red coat, as you see on the right here. Also imagine red-coated cells survive better, perhaps they avoid predators. As a consequence, the red cells replace the brown cells in the population, and evolution by natural selection has taken place. So there's a linkage between the concepts of the cell, concept of the gene, and also this idea here. This is a profoundly important idea in biology, so much so that the 20th century geneticist Hermann Müller used it to define what life is. He said, living things have properties which allow them to undergo natural selection, that is, to evolve. Now, I'm going to use that as the basis of what I'm going to sum up, but I need to do a couple of things first. First of all, I want to say goodbye to Darwin as an old man. Here he is, with the final monumental sentence from The Origin of Species. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Tell me why we cannot write sentences like this today. You can read this sentence here and feel inspired by it. I can't read my own papers 3 months after I publish them. By the time they've gone through the editors and the reviewers and taken out anything of interest or anything colourful, and I wouldn't bear to read it 10 years later, yet we can still read something from this time because it's so beautifully written and is careful. Why on earth is he referring to the fixed law of gravity? Okay, it seems irrelevant in this, just a bit colourful. I think he's referring to the physicist, great physicist Isaac Newton, that biology can have fundamental laws just like physics. Because biology at the time was collecting objects and describing them, and he was projecting laws just like physical laws. So that was one thing. That's saying goodbye to Charles. But before defining life, I need to consider the difficult category of intermediate life forms like viruses. So here's a virus. It's a virus, a bacteriophage. It infects bacteria. They undergo evolution by natural selection, as we saw or see with COVID, of course. Thus, they pass Muller's test that I just said. But they can only reproduce when they are inside the cells of other living organisms. And they do so by hijacking the cell's molecular machinery to copy the virus's genome and to make more viruses. This means that a virus is completely dependent upon another living entity. And that's why it's always given to children at school, is the virus alive or is it dead? Now, in thinking whether viruses are alive or dead, I think it's important to remember that other forms of life are also, to a greater or lesser extent, dependent on other living beings. The many parasites that live on or inside the cells of or bodies of animals or plants or fungi, including ourselves, are clear examples of this. Their extent of their dependencies are less than for a virus, of course, but they're still dependent. Other organisms are used as food. I mean, we eat, of course, other organisms. There are certain amino acids that humans cannot make, and we obtain from microbes that live inside us. Therefore, there's a graded spectrum of living organisms. Different life forms have varied dependencies on other life forms. In the case of the virus, it's very strong. In the case of others, it can be weaker. But I argue that these different life forms should be considered as alive. It's just that the extent of the dependency alters. That's my way round dealing with that. Now, I think the key issue is, just as Muller said, is that they are capable of evolving by natural selection. And this has several consequences. It means that life on Earth is fundamentally connected in two ways, both through being related to each other as a result of evolution, the tree of life, and secondly, through these deep relationships and interdependencies that I've just mentioned. Now, I began by saying I would describe the 5 ideas, which I've done, and I now want to bring them together and summarize where I think we've got to. But the definition is long and you wouldn't find it in a dictionary. Living things are independent, bounded physical entities, so computer life is excluded. The basic unit of life on Earth is the cell, maybe not elsewhere, which is separated from their environment and communication with their environment, so escape the second law of thermodynamics, and also need to produce energy to establish that. And this allows cells to generate order and organization. Multicellular organisms, like our cells, are aggregates of cells. Living things possess the ability to evolve by natural selection or have been produced by natural selection, and to do that they have to grow, they have to reproduce, they have to have a hereditary system which exhibits variability. Living things are chemical, physical and informational machines. They construct their own metabolism, and that is used to maintain themselves, to grow and to reproduce. You can see this would fill up about a page of the dictionary, which is why it's so difficult to get a dictionary definition. Being more specific about life on Earth, we can say that's based on carbon polymer chemistry that produces nucleic acid information storage devices and protein chemical and physical machines. Remember, I emphasized that difference of those chemistries. Life is based on information management, stored in a linear form, encoded in polymers, and life on Earth is related by common descent and are closely interconnected and dependent on other living things. Should we ever encounter life elsewhere in the universe, and I think given the size of the universe that's probable, but whether we discover it is not so probable. I suspect, however, it will be based on rather similar fundamental principles. The chemical details could be different, maybe not based on carbon. The science fiction writers like silicon a lot, for example, and maybe different chemistries will thrive in different places. However, to satisfy the need for long-term information storage, I think it is likely the chemistry will be built on polymers, because that's the way you can store information. So I think that's the commonalities. But I'm going to return to our planet, because that's the only corner of the universe where we know for certain life exists. The life that we are part of is extraordinary in its diversity, but we are beginning to make sense of it. But our knowledge is going further. I've explained how biology shows that all living organisms are related on this planet and closely interacting. And we are bound by a deep connectedness to all other life, to the bacteria, the fermenting yeast, pea plants, the yellow butterflies that I've been talking about during this talk. Together, all these species are life's great survivors, the latest descendants of a single, vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. I want you to consider that sentence, that stretch back through an unbroken chain of cell divisions into the far reaches of deep time. It means we are connected physically through this process of cell division, going back through, of course, to the sperm and the egg from our parents, back through their sperm and egg, and further back to the beginning of life. I think that's something that we don't think about. We are connected physically. I find it extraordinary when I think about that. But as far as we know, we are the only life forms who can see this connectivity and reflect on what it means. And I argue that means that human beings have a special responsibility for life on this planet, made up as it is by our relatives, some close, some more distant. We need to care about it. We need to care for it. And to do that, we need to understand it as well as we can. And if you've enjoyed this talk, I've done it a bit more fully in this book. Thank you very much for listening to me.
Thank you, Dr. Nurse. We are now open for questions. We have time for a few questions.
I go and sit over there, do I? Yes. Thank you.
I want to ask a question.
Where are you?
I'm here.
Let me stand up and sort of... Yeah.
Here.
Got you.
Yeah. And you said that the signal transduction from cell to solution and to cell is a pathway to transduction, like ERG or MEG or RAS-RAF. And how to find these proteins or substance or pathway between cells and solution? And how do we find another pathway or something else?
I think you're probably referring to my suggestion about wetware, or it wasn't my suggestion, it was Dennis Bray's, that when you have solution chemistry, when you have different components, signal transduction, as you said, connected to each other by solution chemistry, then you can rewire them in different ways, because if A is interacting with B but can potentially interact with C, then you can change the way the pathways work. Now, that has been shown by biochemistry, to answer your question, in certain cases. But I have a feeling, and it's only a feeling, that it will be more prominent than we understand, because we tend to think of everything in linear pathways. There is evidence of bifurcation, and then there could be evidence for rewiring as well. And that introduces an extra level of ability to generate complexity, which is obviously what we have in a living cell. I don't know if that answers what you were thinking about, but the different mechanisms by which it's produced, I could imagine them, but they have only been described in a very few cases.
Okay, thank you.
Thank you very much. I'm interested in why would you exclude computer lives when you constantly make analog to the circuits and computer, whereas, and as you mentioned about the circuit-based lives, and it's one of them, and then how about those organisms that don't reproduce? How would you fit that into the evolutions? How do they do selections in other ways or actively in different forms? Thank you.
So I think you're thinking about several things there in the metaphor of the computer, of course, in thinking about complex systems. And maybe you were touching upon, but didn't quite mention whether computers can have some sort of life within them. I don't know if you were thinking about that.
People arguing about that.
Yeah, exactly. Well, the first thing is, I think we're a very, this is to generalize your question, I think we're at a very exciting crossroads in biology, where the chemistry and physics generated largely by molecular biology can now be combined with more sophisticated management of information, for which computing is very helpful. And we need to fuse those together. It isn't a trivial problem when people try to do it, like writing 1,000 differential equations to describe all metabolism. I think that is not the way to go about it. But the ability to somehow put together information together with chemistry and physics, I think is the next big step for biology. Now, computing is a metaphor. It is useful for thinking about it. It's a crude metaphor, but it is still, we're still thinking about it completely crudely. So I really think there are really strong opportunities in linking those things together, which I think was the gist of your question. And it's been talked about for quite some time, systems biology, since I was an undergraduate. We haven't made much progress there, to be quite honest with you, but I think now we've got to take a step back, and if I can be a critique of what we're doing, with the chemistry and physics, we are endlessly describing lots and lots of behavior of molecules in cells, thousands, hundreds of them, and we publish papers which describe but don't lead to understanding, if I can put it that way. And I think we're only going to get understanding if we try and put this together in a much more sophisticated way. And although you didn't ask these questions, I'm now on a roll a bit on it. The way I try to address the problem of cell cycle control is actually to take the networks that we have and simplify them to a level where they still work, and then use the simplified system to try and establish the principles that allow us to go back to the complexity, if I can put it that way. Because if we try and deal with the complexity when there's redundancy and so many components, all we end up doing is describing behaviors without understanding. If we strip out components and still get it to work, then that will lead to principles that we can actually apply. You were nodding your head there. Did you want to come back with something?
No, I'm glad that I'm taking similar approaches in solving these, but well, the second question about evolutions,
Yeah.
like cells that don't reproduce or like sexless reproductions or clones, and how do you put that into the evolutions or like the selections?
Yes, so evolution, this is an important question. I'm going to divide it in two ways. What we can do easily is describe the different components and how maintained they are through evolution. And we tend to do that. So in the introduction you heard I isolated cyclin-dependent kinase in fission yeast and then showed the human gene could substitute for it, and showing that the whole thing was highly conserved. So evolution allows us to describe the components easily. What it isn't so easy to explain is whether the system itself within which these components are embedded is also conserved or not. And to some extent it obviously is, because the controls that we worked out in yeast actually are relevant to complex cells like our own. But it could be that there's a robustness in certain types of control systems, which are also evolutionarily stable, as well as the components within them. Am I making sense here? So it's not just the individual components, but the way they are linked together. And I think that is a step that we now have to explore more, about whether there are certain control configurations which are much more stable and much easier at developing outcomes. And I'm here on the edge of understanding, but I think that's what I personally believe, and I think we've got to think about conserved system controls as well as conserved molecules. This has got very little to do with my talk, I have to say, but it's perhaps relevant. Yes.
We have another question. Yeah.
Yes.
Just a quick question. I thought it was absolutely
Do you want
fascinating.
a quick answer then as well?
Yeah.
Rather.
Yeah. So, have you seen very many parallels between biological evolution from DNA information and digital technological evolution, and like automobiles evolving different models and different computer systems? I think you hit on that a little bit, but have you seen many parallels between both types of informational evolution?
Well, this touches really on what I was just saying.
Yeah.
And I think I'm talking in metaphors, and we have to be careful about metaphors, but I think it is a good starting point, because you're absolutely right. You look at a whole wide variety of cars, and they're all built on the same sort of structural way of connecting things. And in a way, if we keep the metaphor of the car, for a long time we just had primarily internal combustion engines, okay, but then we've replaced the internal combustion engine with an electrical engine, which is completely different. But within the system, it performs the same role. So if you just describe the petrol engine and all the components to it, you don't emphasize as much as you should, the contribution to the system as a whole. But if you now think we've replaced it with an electrical motor, which is completely different, and they do overlap in power, but within the system they perform the same job. And that, I think, is still a metaphor, but one maybe that's worth exploring.
Thank you.
Thank you for a wonderful and insightful talk. You mentioned that all cells come from cells.
Yes.
What if we can synthesize a cell?
Would we consider?
Would you still consider a synthetic life?
Yeah.
We can now synthesize all DNA, and I presume can synthesize the necessary protein to reproduce and wrap into the lipid.
Yeah. Well, this is interesting, isn't it? The first thing is just let's think, if we take a cell out of your body or my body, we can grow it and it will have the properties of life. It will not have the same properties of life as we do as a human being. It's got a more limited set of properties, but it's still basically alive. So we've got to, the reason I mention that in this context is just to be clear, we're not saying a human cell is alive like we are alive. It has more limited properties, but it does have properties of life, but just not the overall complexities that we have. Now, then the question you asked, though, was about, I think, making a synthetic cell. And it's... I'm not sure how we could make a synthetic cell. I'll be quite honest with you. Some scientists who, like the media, say we have created life when they've created a DNA molecule, and I mean that's just bullshit, frankly. So I mean, it's just good for the newspapers and not much else. And we throw newspapers away every day, as you know. So, but what you have to have in a cell is all these structures which are actually made within the cell itself. So I suppose you could purify the different components and put them together, but I ask myself, what's the point in really of doing that? Because one of the wonderful things about life is it is constantly making its own components. And in a way that a car doesn't, for example. I mean, you're constantly... So you as a human being are completely different in terms of the molecules in you than were 10 years ago. There's probably not a single molecule the same. Yet, we look at you and you're the same. Well, a bit older maybe, but I mean, it's the same. So there's some extraordinary things going on. And if we are challenged by making synthetic life, how on earth did life arise in the first place? Because you have to have all these components working. You need to have a genetic system. You need to have it within a cell, and it needs to be connected. You seem to have to invent too many things at the same time. So it's a mystery to me. I do think about it, but it is a mystery. That was a lot of words to say. I think that I don't think we could make a synthetic cell from scratch. I think we could make a synthetic cell with the different components, maybe, but I don't see the point in doing that. But really, because but it is a very important question because it makes us think about the origin of life. And I haven't an answer to that.
We have some young audience in the audience. So there's one, and there's two, and there's one there. Is there any young audience that have any question? The younger audience? I'm looking at you, the two in the middle.
I'm curious that is the signal transduction pathway in stem cell the same as the differentiated cells?
In principle, yes, but different components. So the principles are the same, but not the particular molecules.
And how do we control the stem cell proliferation rate?
Don't know, don't work on stem cells.
All right, let's move on to the next question then. Any youngster? Oh, okay, yeah.
Hi, sorry. I've been thinking about this question for quite a while as well, and
Where are you?
I'm here.
Gotcha.
Yes, I'm quite curious about your opinion. Considering that you are exploring the universe and on another planet, and you run into another interesting, let's say, material, how would you use the criteria that you just defined to argue that the thing you run into is actually alive?
Sorry, I didn't quite catch that one. Did you catch that one?
Sorry, I wasn't listening. Can you repeat again? Sorry.
Yes, I'm quite curious about your opinion in this situation. Assuming that you are exploring another planet that is not on the Earth, and you run into a certain thing that you are not sure whether it's alive or not, are you able to use your criteria that you list in your?
Are you asking a question about when is death?
No.
Mm?
A different planet. How do you define life in a different planet if you don't know much about it?
Well, I would use all the words I put on my last three slides. I think for me, life on another planet should be, core to it is a system that can undergo evolution by natural selection. And that requires an informational component, and it requires heritability of that informational component, which is variable. And that informational component is determining the metabolism, that is the chemistry that goes on. All of those would be, I think, need to be put together to define life on another planet, even if it was based on silicon or whatever. Now, if you were a space biologist, that would be looking into the depths of space for other life forms. That would not be a useful definition. The only way we can really look at distant planets is by looking at the chemistry of those different planets, and we are then asking questions usually about the chemistry that is life as we know it. So NASA looking for it will be really, the experiments will be determined by life on this planet. If life had a different chemistry but the same systems connections, we wouldn't detect it. So what I'm saying is, the way we're defining life is more as a system than as particular molecules, but the way that we look for life is through particular molecules, particularly carbon polymers. Does that, you see the problem, I think? Yeah.
So I'm going to ask a question, and maybe during my question Q&A, I want to invite the younger audience, a few, I think middle schooler in the audience. If you have any question, if you don't know how to ask in English, we can translate for you. So my question is that you use very simple language to elegantly explain how complex cell processes work. And you talk about evolution as being sort of design without designer,
yeah,
which is a very interesting thought. So my question is, I think in the industrialized sort of society, the change seems to be too quick for the evolution to pick up. So we all know some of the consequences. And so what's your view on how do we deal with it? Of course, we talk about climate change and what we can do, but practically what can we do to, sort of harness what we know and preventing bad things from happening?
Well, it's a pretty difficult question, of course. I think, so I'll answer it in rather vague terms. I think we have to develop a respect for what we find around us for life, actually. And it's extraordinary how it works. And we have a biosphere on this planet which does work. And it could be more sensitive than perhaps many think. And you mentioned climate change, and if we have significant climate change that makes pressures on life too quickly changing, we cannot evolve by natural selection to deal with it. Now, I doubt if all life will be extinguished there, but it could be quite a lot of the life as we know it would be extinct there. Which is why it's so irresponsible when political leaders like Trump, for example, simply rubbishes it, because it shows no leadership. It shows that there's no, we shouldn't be thinking about the future. We should only think about the next 3 years and whether we can, make some money on it. And so we need proper, mature leadership that recognizes the frailty of life and the importance to protect it. And that requires a different sort of long-term thinking in politics, if I'm allowed to be political in the academy.
All right. Noah Youngster, you first, and then
Thank you, sir. I thoroughly enjoyed your talk. My question is that do you have biological or evolutionary or information chemical-based explanation for conscious?
Because
conscious.
Because after all, Homo sapiens, we are the billion-year result of evolution. But so far, we are the only species to make cars, ships, and rockets and go to the moon. Thank you.
I'm not sure going to the moon is a result of consciousness, but I mean, just putting that aside. Yeah, I've avoided trying to explain consciousness. I go to talks about people talking about consciousness, and I go away none the wiser, usually, as a consequence of it. I assume that's my own weaknesses, but what I would say is consciousness is central to it is self-awareness. And living organisms, even cells, have some sense of self-awareness. And I would say that that other more complex living organisms, like mammals, or for that matter an octopus, has self-awareness. So when I ask questions about consciousness, I sort of retreat a bit and say, well, self-awareness is the beginning of consciousness. And I would start there, because if you're aware of yourself and your sense in the environment, I think you have some sense of your own self as an individual, and that has reached a more sophisticated way in for example, a human being. But I suspect the beginnings of it are found in other living organisms as well. But I do struggle with it, I have to say, that how this sort of grey matter up here can lead to our sense of identity, character, and everything that we are as human beings does seem extraordinary. So I don't pretend to you that I've answered your question, but I would at least start with self-awareness.
We take 2 more questions, or 3 more questions, 1, 2, and 3. You first.
Okay. I have read a paper with several anomalies about twins against Darwin's natural selection. Since those twins have the same mother and the same genes, but one is handicapped and the other is in normal physical conditions. So what's your methodology or attitude shall we apply to solve this anomaly? Is there any hypothesis or Schrödinger's concept to solve this anomaly? Thank you.
Yeah, I didn't quite get that one, but...
So he's asking if they're twins, the same identical twins.
Twins, yeah.
And there's one has some disability, the other one is normal.
Yeah. Well, I think the answer is straightforward, is that you are a product of your genetics and your environment. And when twins are separated, then we have seen differences. And even twins brought up together will have a different exposure to the world. So I'm less troubled by this than perhaps many people, because although I'm a geneticist, I'm not a strong genetic sort of determinant. I always, when people say, well, is this due to genetics or environment, I say, well, my starting position is 50-50. And then with certain traits, like the inability to for an enzyme to make a certain amino acid or something, is completely determined. But then, of course, you can replace it with something from the environment, from the amino acid. So then it becomes from genetic determined to environmentally determined. So you just have to be careful about how you do it. But I think twins, I feel we can understand, because it is a mixture of genetics and it is a mixture of environment.
Thank you for your talk. So my question is, there are many different levels in biology, and when applying the concept of evolution to each level, do you think that there is some sort of scaling concern? Like, are all the levels scalable, or are they not? And the second part of the question is, do you think that there is a prominent level, like at the level of the cell, or the gene, or the tissue, the organism, or even the ecosystem?
No, it's an interesting question. So when we're looking for explanations in biology, we sort of move up complexities. You start with molecules, you go to cells, you go to organs, you go to an organism, you go to a population. Now, what I think about that is that the systems part of this is it may well have commonalities across all of those categories. In other words, each of them are a complex system. Molecules work together in a complex system. If we go to the very top, a community, a population, individuals are acting in a complex system. And I feel that sufficiently strongly to say that we can learn from each other in those different systems because there is something similar about them all. The second thing I wanted to say, which isn't again quite what you asked, is that each of these levels influence each other. So if you have molecules, they influence cells. But when you have the cells, they actually also influence the molecules, so that there's a sort of feedback from that higher level of complexity. So when you study molecules in the test tube, you don't get the full picture, because the molecules act within the cells, and the cells themselves restrain how the molecules can actually behave. And it's like that all the way up. So 2 points I'm making. One is the systems part, not the components part, is similar at these different levels, at least in part. And secondly, at the higher level, they restrain lower levels. So proper biological understanding requires investigation at a number of different levels, is what I'd say in short.
One last question.
Hi. Yes, thank you, Dr. Paul, nurse. Since you mentioned about the virus, I have a question about the COVID-19. Since around 7 million people died in this kind of disease, can you explain or elaborate from the point of evolution and your philosophical thinking about this kind of event to explain us if that is a bioterrorism act or it's just a natural pandemic? Thank you.
Again, I think I might...
I don't, and I couldn't see the...
Let me have a go at what I think. I mean, we're talking about talking about COVID, okay? Yes? And I think you were commenting on how it evolves, essentially. And I'm not an epidemiologist or a virologist, a medical person, but what I would emphasize there is that when you have a virus and it is spreading from human being to human being, if it is very virulent, it won't spread because it kills before it can spread. So nearly always these viruses evolve to be less toxic as you go on, and we've seen that with COVID, because if they are too toxic, an individual dies and they can't spread. So there's different strategies. Viruses can remain latent for a while and then so they can spread. But you've got to always think about what is the effect of the virus on the body when it's infecting many cells and therefore disturbing the body, versus the ability of that virus to spread to other human beings. So that's got to be factored into the evolving situation and certainly applied to COVID, which is why COVID basically got weaker, and as the virologists sort of predicted.
Is it allowed one more?
Yes, of course.
Yes.
Hi. So I have a question over here. For the natural selection to work, you need diversity, and that means you need mutation. However, when cell replicate, they actually had the checkpoint to make sure you don't really make mistake. So how does this balance work? Can you comment about that?
Well, I think there is a balance, because if you have too much mutation, you will the system is prone to disintegrate. But if you don't have enough mutation, you don't evolve. And it's quite interesting because one way of producing mutations is by altering the DNA polymerase that copies DNA. And you can get mutants in bacteria where the DNA polymerase is better than the normal one you find in a bacterium. And they are at a disadvantage because they cannot actually evolve fast enough to deal with changing situations. So although at first sight you would think that such a system should be perfect, if it was perfect, it would lead to extinction. And it's a balance. The balance is achieved by what survives. So if it's too perfect, it will become extinct. If it is too imperfect, it becomes extinct. But there is a balance in between where it seems to work.
Okay, that's all the time we have. Let's thank Dr. Nurse again for the wonderful
Thank you.
talk.
Thank you.